Saurabh, sorry, we're having trouble with your audio. Saurabh? Saurabh, can you hear me?
Ranging dogs. Like
Oh,
they are diverse, since mostly human-made. The diverse location in the human house, on the roof, in the food. Yeah, these are diverse. So these three, and try to understand that difference in the things or not. So how with this uh you know labs or the same say suit of ranging, these actually give us uh, this they have interesting preferences because
Saurabh? Oh, shit. Saurabh, can you hear me? Saurabh? We can't hear you. Can you, in the top where it's asking you to accept sharing, can you click OK? Your audio hasn't come in. Um, can you speak now? Can we hear you? Try again. We have totally lost your audio. Can people hear me? Yeah, okay. Um, s- Saurabh, do you have a message at the top of your Zoom asking you if you're okay with the recording? I'm very sorry about this. Oh, where'd you go? Okay, I think he may have logged out and be logging in again. Sorry about this, folks. Um, of course, his audio cut out right as we started the talk. He was fine before. Um,
hi.
yep. Hi, sir. Oh, there you are.
Okay, okay. So I wasn't able to hear you guys. So was there any problem there, right?
Yeah, so we did lose your audio. Um, hopefully you're back for good. Let me see. I've got to make you co-host again. So
Okay.
if you
You can hear me,
can,
right?
we can hear you now. Yeah,
Okay, okay, fine. Fine. Let me share again.
yeah,
So, can you tell me where we were actually? So it will help me to. Uh,
yeah. Well, you've got about.
this this is
s-
something can you like when you lose my voice like here, here, here.
We lost your voice right at the beginning. Um, uh,
Okay, is it?
yeah. So we've got about 6 minutes left. So are you able to do like a lightning?
Okay, okay, fine, fine, fine. I'll just do that.
Okay.
Okay, fine. So thing is that we were trying to understand the resting preference for free-ranging dogs. As we know that this resting is a survival and crucial behavior. So there are certain things that can actually influence the dogs to rest at a certain location. So so how we have done that? So these are the different resting sites we have definitely looked into, and the thing is, this is a particular study that actually helped us to um, or to being curious that whether resting the free ranging dogs has a resting preferences or not. So how we have done this? So our primary area was that how we have done that. Okay, so the resting site preferences for that. So usually I'll just keep those. This is a pan-India projects. We have done that with 66 groups and 284 dogs, and we've done in a round clock and these things, and we've done for the 4 different seasons in 2 years. So how we have done this? So this located groups in sending a scan we have done, and also had a GPS location of that particular area, and as well as note down the physical properties of that particular things. So thing is, there are several physical properties that we are actually considered. So what about those? So the either particular resting site has a shade or not, the intensity of the light, the weather of the particular areas, the different weather parameters we have considered, the disturbance of the resting area where they're resting, the presence of insect, distance from the food, distance from water, the surface where they're resting, the level they're resting, the visibility they have, or the position of the territory and the place where they're resting, kind of, and like they're resting in the side, the roadside, in the road, or in some anthropogenic structure or not. Fine. So how we have done this? We had different set of behavior. So resting for what we have determined is that resting is a kind of a state where we would let them know that they are either lying down or sleeping. So and the different behaviors we have encountered, we have categorized them depending on the different on the amount of energy they are conserving. So slightly relaxed can be, say, they're in a resting state but watching, or they are alert. Moderately relaxed, they are lazing, gazing, or the grooming itself. And extremely can be they are sleeping. So what the other things we got to know. So out of all of these factors that was going to affect the resting size, so to understand that, we run our ordinal logistic mixed model where we try to understand the quality of rest, like from the less restive, from the moderate restive, and the highly restive, how that has been impacted by the different factors. So what we know that near the food dogs, the resting quality increases. So as in the low disturbance areas, in low to moderate light condition, in a shady area, they rest more. In roadside and nearby human, the resting quality increases, as well as in the center position of the territory and with high visible condition. Fine. Then we try to understand, fine, with the 7 different factors, that those are important, but we wanted to deep dive into it. Like, fine, sevens are important. Do they have most important things, like out of 7 that are the most crucial thing, without that they won't rest in a particular area? Fine. So what we have done, so we try to understand the proportion of time they've rested in different categories, and we have and we done this analysis in different state of this of the dogs, like the restive, moderately, and highly restive one. So let's say these are the free-ranging dog habitat, and what are the different factors that is important? Fine, we found that territory position is the most crucial thing, one of the most crucial thing. Then the distance from the food is the another crucial components for the decision to rest in a certain area. Then the visibility. So these three are the most important components. Without that, the dog won't rest in a certain area. Fine. We try to understand in which territory position. So these are the edges area around 5 to 10 meters in around their territory, the edge. 10 to 15 meters radius called the center position of the territory, and rest are the intermediate. Fine. So then we try to understand which are the most preferred ones. So irrespective of the resting state, dog always choose the center position of the territory. In the distance from the food, those were less than 2 meters of the food, 2 to 10 meter. Another category was greater than 10 meter. So what was we found from there? We found that irrespective of the resting state, dog will always choose to rest nearby resources. Well, what about the visibility? The high visible condition. So these were high, they're resting the surroundings area, all the four sides are open and they can look into every direction. If at least two directions are blocked, the moderate one, and if three of them are blocked, it's called the low visible condition. What we know, dog will always choose the high visible condition irrespective of their behavioral state. Fine. So make it short, but the things we want to conclude from here. Fine. Dogs, they always try to rest near the resources, center position of their territory, and in high visible condition. Fine. So if you find a free-ranging dogs in a particular habitat, what you can predict? If it's resting, so it should rest close to its resources, center position of their territory, and a side where there is a high visibility. Okay, so that is the takeaway home message that we try to understand that has the free-ranging dog has a preferences for resting. These are archive currently right now is under review, and I would like to thank Anindita, Kalyan, Kaushiki, Maya, and Laya for making this work happen. I'd like to thank Third Joint Congress Evolution Biology virtual version for giving the platform, SSE for giving and helping me to participate in this particular platform, EGC as a Kolkata and Build Lab members for their constant support. Thank you. I think if there is any minutes left, I can move with a question.
Thanks so much, Saurabh. And thank you for rolling with the tech issues. We'll move on to the next speaker now. If you have questions for Saurabh, you can use the chat. And we'll move on to our next speaker. Oh yeah, yes, Tim.
I think we can use the Q&A that will also be available for the recordings later on.
Okay. Oh, perfect. Right. Yes, you're correct. Okay. Thank you. Okay, Sazmit, did you want to? So our next speaker will be Sazmit Naik from Cornell University. Okay, and we'll just wait. Oh, and you can get going whenever you'd like.
Okay, perfect. Am I audible?
You are.
Okay, great. Hi. So I'm Sazmit Naik, and I'll be presenting the work I did for my master's thesis today when I was at University of Montpellier. My thesis title was Evolutionary Dynamics of Thermal Adaptation During Range Expansions. And okay, so to start with, when do range expansions occur? As formerly unsuitable habitats become suitable, species are likely to shift their ranges in order to track the climate. And forecasting of species distributions can be pretty complicated and traditionally has been done by using abiotic properties of a species' current habitat and using it to infer properties of a suitable niche, and which is used to predict future ranges. But local adaptation to newer climate regimes remains absent from these models, especially because data can be severely lacking. But that is not to say local adaptation doesn't happen. An example of an active migration is that of the damselfly in Sweden. As with climate change, temperatures in the native habitats have been rising, so they have been seen to be moving into colder northern ranges and experiencing changes in their growth, development rate, and feeding rate. Then though theoretical models have been used to understand impact of local adaptation on range expansions, thermal niches have been modeled using a symmetric Gaussian niche with like linear flexibility. And often thermal niches have constraints on how plastic they can be at different temperatures, and that comes from underlying biological constraints. So the question we hope to ask is what is the effect of considering different thermodynamic constraints on thermal adaptation during range expansions? Okay, so I'll now introduce a basic structure of the model that I've that I've been using. Temperature is, as you know, an important biological rate modulator. Its effect is interesting, but it's also difficult to study as they have scaling effects, especially in ectothermic species. The body temperature can affect rate of protein reactions, which scale up to the metabolic rate, and that is strongly associated with feeding and foraging rate, which in turn leads to temperature dependence of the population dynamics, that is the birth and death rate. And finally, that affects range expansions and range expansion dynamics across the temperature gradient. And what I hope to introduce is a simple model that covers this whole levels of organizations. And this here is the simple structure. Okay, so I hope to model metabolism as a function of temperature and consider feeding rate as a proxy. Thermal performance curves generally across levels of organization have a characteristic shape. The first half of the curve is exponentially rising, and that is often attributed to a thermodynamic effect, where increased temperature leads to increased collision rates and therefore increase in reaction rates. And this first half is what has been used to model effect of temperature on biological rates. But what is often like not taken into account is the second half of the curve, which is the skewed peak and the steep fall. The mechanisms underlying this may not be as generalizable or well understood, but they play a pivotal role in understanding adaptation at higher or extreme temperatures. So what are some of the hypotheses that are used to explain this sort of fall-in activity? One of the hypotheses from literature has been that there is failure of essential enzyme reactions underlying metabolism. And second has been the failure of protein folding or denaturation. And though these two mechanisms seem similar and linked, they have different protein properties under selection and therefore lead to very different thermodynamic constraints during thermal adaptation. And I wish to try model them into a thermal performance curve and look at its scaled effects to range expansion dynamics. So we did bit of a literature review to look into models that have been used to describe temperature dependence of protein reaction rates. And I won't be going into the mathematical details, but give sort of a biological summary for the three models that I'm using. And so first I consider the temperature dependence of enzyme reactions, and this has been described by the macromolecular rate theory. So let's consider a thermal performance curve. And for the thermal performance curve to stay positive at higher temperatures, the enzyme substrate binding needs to happen successfully. And this requires the enzyme to be able to change its conformation and bind like strongly to its substrate. At higher temperatures, enzymes have the tendency to become more flexible, and adaptation to higher temperatures include making enzymes more rigid to increase binding strength. As a result, enzymes staying functional at higher temperatures, they lose flexibility to maintain function at low temperatures. And for those interested, this phenomenon is known as the enthalpy entropy compensation. So that is one route. And another route of adaptation is by changing heat capacity difference of the reactions. This is a more abstract term. And what it does, it controls the rate with which the rigidity of enzymes change with temperature. And changes in this parameter lead to this sort of breadth versus height trade-off. Okay, so that's my first model. The second model is based on the second hypothesis of temperature dependence of protein denaturation. Here, as temperatures increase, the rate of activity increase, the number of folded protein copies decrease, and therefore overall the metabolic rate also decreases. And one of the properties under selection is the entropy change accompanying protein denaturation, which is similar to the flexibility stability trade-off. More flexible proteins are seen in cold-adapted bacteria, which allow for activity to occur at lower temperatures, but they quickly denature at higher temperatures. And the second protein property of stability is considered, where higher thermal stability allows for slower denaturation of proteins at high temperatures, with not much change in rates at lower temperatures. And finally, as a control, I consider a Gaussian curve with asymmetric flexibility and where the mean and variance can have linear mutation effects. And what I do want to is hope to link these different flexibilities in thermal performance curves to population level effects. Okay, so I introduced these three models into an agent-based model of range expansions with a linear landscape and a linear thermal gradient. The population is initialized at the center of the temperature gradient, and the individuals can expand to either ends with a cold and hot front of expansion. each individual's traits determine their birth, death, and dispersal rates. And an individual's protein properties define the temperature dependence of the overall growth rate, and that is according to the 3 discussed models before. So overall, it results in a roughly unimodal spatial distribution of the growth rate across the landscape and temperature gradient. And an interesting emergent property is the unimodal distribution of the carrying capacity across the landscape. And this is because of the scaling of the feeding rate leads to scaling of the interspecific competition, and therefore leads to this sort of non-linearity in the carrying capacity. And here is an animation of the simulated range expansion for one of the models without any trait evolution. The top panel shows the population density on the y-axis, and the x-axis is the linear patch landscape with the linear thermal gradient. And the population adapts to the center 10 patches for 2000 hours, and they're allowed to expand to either sides. And the top panel shows the population density, and the second panel here shows how the growth rate shows how the growth rate changes across the landscape. And so finally, here is the sort of the differential equation that captures the dynamics if there is no trait evolution. The first 2 terms here show the birth and death rate in the xth patch, and the rest of the terms show the dispersal into and out of the patch. And so we have these bunch of coupled ODEs, and we stochastically simulate them using the Gillespie algorithm. Okay, so finally we come to the results. So what we want to do is compare the average range expansion dynamics, and for that we use the patch front. Patch front is the furthest patch with non-zero population. And the y-axis here gives the patch front, and the x-axis gives the time. Sorry. And so we initialize at the center of the landscape that's around the 60th patch, and that leads to a hot and cold front. Here is the simulation case with no mutation. So the patch fronts stop when the growth rate becomes negative at extreme hot and cold temperatures. And we also showed the speed of expansion using by fitting a linear graph to the initial patch fronts and plotting the slope for the cold and hot front speeds. And here we can see that the cold front speed is slightly higher than the hot front. So next we look at the Gaussian model. Okay, so we also look at the Gaussian model, and we see that it goes a lot further than the non-mutation case and shows more symmetric speeds. But finally, when we look at the thermodynamically motivated protein denaturation based model, we see a complete skew in the trend with much hotter, like hot front speed versus cold front speed. But what's interesting is that this does not hold when we look at the enzyme catalysis based model, where we find trends that are much more similar to the Gaussian case. So after this, we basically did a bunch of simulations and sensitivity analysis to understand why we have this kind of variation in range front speeds. And our main observations have been that in the protein denaturation based model, because of its hotter is better trend, it is able to actually have an accelerated expansion towards the hotter gradient. And but that is not the case for the enzyme catalysis based model, which has a strong trade-off between the rate of reaction and the binding strength. And that leads to a much faster expansion along the colder gradient, the same as the Gaussian curve model. And this happens because when the adaptation is slow, the carrying capacity makes a important difference. So along the hotter gradient, because the carrying capacity is smaller, there is a lot more stochastic patch extinctions, which slows down expansion. But along the colder gradient, because there is a higher carrying capacity, it allows for a faster expansion. And another observation that we have had was that because of rapid adaptation of the protein denaturation based model, standing trait variation at the edge is much more reduced compared to the slower expansions that maintain a much more standing trait variation. And so with that, I sort of come to the end of my results. And the implications of the study is that when you consider the mechanisms of adaptation, as we have in this case, especially because these mechanisms have been seen in many thermophilic or thermophilic bacteria, like these different routes of adaptation, they can make a difference when we're looking at the actual range front dynamics. And going back to our introduction, a recent study on the damselfly has shown that there's been a change in the fraction of metabolic alleles, metabolic protein alleles across the thermal gradient as they're expanding. And that just serves to prove the point that considering the micromechanism under adaptation can be important to understand the infant dynamics. Okay, and so with that, I come to the end of my talk. I would like to thank my advisor, Dr. Emmanuel Fraunhofer, and my team at University of Montpellier, and my undergraduate institute for funding. Okay, I can take questions.
Great, thank you so much, Seismi. Really lovely talk. Do we have any questions? We've got a couple minutes. So I was wondering, I mean, like, I think it's a really nice sort of modeling framework you've got here. Are there some clear sort of experiments one could do to sort of to help refine your models, or to sort of explore where you would want to go next?
Yeah, that is actually, yeah, that's definitely true. I think definitely my model is more pertained to like smaller organisms. But in a lot of them, there have been clear like sort of important metabolic proteins that have been recognized for like say carbon metabolism. And I think yeah, you can do experiments to just see what, especially in artificial selection, that what are the proteins that are how like major metabolic proteins changing their shape to like actually yeah, to adapt to higher temperatures. But what is important is that each protein, it can be super different. So the fact that that the same mutation affects the binding strength and rate of reaction is not necessarily true. So it has been seen for a lot of enzymes, it's not necessarily true. So it depends on the structure of the protein whether or not you can independently change rate of reaction and binding strength. But
yeah. Okay, no, thank you so much. I appreciate it. I do see a hand raised, although we do have to move on to the next talk. Henry, if you want to put your question in the Q&A or the chat, that conversation can continue on the side. So thanks again for your talk. Our next talk will be a pre-recorded one. And so while Tim gets that going, so this is a talk from Regine Claire Manguimot at the University of British Columbia. And we'll get that started momentarily. Yeah.
I need to share the sound. That's very important so that we can see.
Absolutely.
Okay, you can see that?
We can, yep.
All right, then I'll get started.
Groups that contain meiofauna represented.
And we have sound, so that's good.
Yeah. Hi, everyone. My name is Regine, and I'll be talking about synapomorphies and trait evolution in small animals. I like to start with this fun little picture to get everyone in the headspace that I'm in, where I look at really small animals. When we think about what animals look like, they look a little different to me. The first things that come into mind are these small interstitial animals known as meiofauna. And some of them are known to us probably, like nematodes, people who study C. elegans, as well as tardigrades, which are these really cute water bears. But there are also groups that are already known that have meiofauna, such as annelids shown here, or maybe groups that are lesser known to us, like gnethostomids and gastrotrophs. What I mean to say is that these small-bodied animals are sort of pervasive in animals and metazoans. So as shown here in blue, we have all the major groups that contain meiofaunal representatives, and in pink, we have groups that are entirely meiofaunal. And I think we'd be remiss to leave them out of our considerations for studying animals, because they're a huge part of animal evolution, even just seeing how they're spread out in the tree like here. The group that I study, and I like to point out, is sister to all bilaterians. So we can see here that bilateria is a very successful group with a lot of the extant major groups falling within it. And the group that I study is the sister to all other bilaterians. And they are known as aseals. So what are aseals? They are flatworms. They're quite small, and they're about 400 nominal species that are mostly marine, not almost entirely. If you were to pick them out of a lineup or a sample of these small animals, you notice that they're the ones swimming quite gracefully. They're entirely covered with cilia, and they have a status that basically tells them which way is down in the water. And they are found in all sorts of waters. They're globally distributed. And as you see here in this global map of known species records, it's not uncommon to see this pattern where Europe has a lot of the diversity characterized as shown here. Coming from North America, I like to make this East versus West comparison. It's a little embarrassing that the Pacific side, which is known for marine studies, is quite low in its record. And then my national pride takes a total hit because there are no records so far published in Canada. But that's not because the seals aren't found there. This is one of my sites in British Columbia, Canada, on the West Coast, and it's actually teeming with seals. And one of the reasons I wonder why they're neglected is maybe because they're difficult to work with. And the reasons I've heard are that there's few character traits, there's not really synapomorphies, it's only molecular, and a lot of the literature is old and there's no molecular data, and all we have are like line drawings. But even without molecular data and line drawings, it's perfectly sufficient to allot them to known species descriptions as shown with these micrograph images I took. They're quite high quality, and you know they're enough for you to connect them to known species from line drawings. But there are also lots of species that haven't been discovered before. And as you can see in this 18S tree that I have, maximum likelihood tree on the left, I have allotted 8 new species, at least molecular wise. And they're found all across the major groups of aseals. And to me, that means that we can study aseal evolution throughout the entire group, even in just a couple sites in British Columbia nearby to me. And so these are the major groups. And as of right now, the taxonomy is largely based on molecular data, which can be difficult for studying evolution in terms of synapomorphies and what might have led to these radiations. So I show here in the major groups, the thickness of these bars are roughly correlated to the number of species in each of these major groups. This largest one, convoluted, having about 120. And I think it's a shame to not be able to explore why there's so much species diversity in this clade here, without looking at evolutionary traits and morphology. Luckily, though, in this group, which is fast evolving, really diverse, aseals known as musculata, they do have a synapomorphy, which is shared ventral crossover musculature. Now, you can't really see this trait unless you apply fluorescence microscopy. I'm going to let our eyes adjust here. But basically what I use is fluorescent tags, this time focusing only on musculature, to see muscular features in aseals. And I love starting with this example because on the left here is what it looks like, this juvenile with basically no identifiable traits aside from its status, cyst, and cilia. But when we fluorescence stain DAPI, so for nuclei and muscles using phalloidin, it lights up really well, showing all these other traits. And the one I want you to pay attention to is these crossover muscles here. These are muscles that have been stained and photographed in the stack. And in blue, we have the most ventral stack, and going towards pink more dorsally. So we can see here that it does have ventral crossover muscles. And this is still useful for mature specimens as well, where you can see a paired ovary situation even before you stain. But when you do stain, you get to see these ventral crossover muscles, and then you reveal this massive gaping hole, which is the mouth. Pretty important as well in terms of character traits and understanding overall how this animal works. So the 2 examples I've shown so far are of 2 species within musculata. They both have those crossover muscles. But, you know, to ensure that this isn't a plesiomorphic trait, I took a look at a species outside of that clade, which is known as Paratomella here. You already see that it looks quite different from the other 2, just even superficially. It's a really cool species that's regenerative because it often asexually buds paratomy to reproduce. It's very weird already, just looking at it, it's very long. And when we do look at its musculature here, phalloidin stain once again, with fluorescence microscopy, there's no crossover musculature on the ventral or dorsal side even. So using fluorescence microscopy and targeting particular features, we can test synapomorphies that exist, or maybe even look for new ones and uncover features that are previously unknown or invisible to us to support or challenge molecular bases for these groups. And yeah. And my future plans is to apply this to nervous system architecture. And I'm going back to the fact that these group A seals are bilaterians, but sister to all other bilateria. We can use it to examine what the bilaterian ancestor was like, or what happened when bilateria came to be, and look for hints as to why bilateria is so successful and extant today. Because at the stage of bilateria, the nervous system centralized from being nerve net to showing nerve cords, which I think allows for faster synapses and coordination of things like active predator and prey behaviors. And secondly, that group Crucia musculata, which possessed most of acial diversity, is also known to be a fast evolving group. So it's already interesting to begin with. It happens to also have very diverse architecture of their nervous system. So I think it would be really interesting to see whether or not that has a role to play in the diversification of that particular group and its fast evolving rate. So with that, I thank everyone for coming and listening. If you have any questions, please don't hesitate to contact me. I'd like to thank my collaborators on the right and answer for funding my research. Thank you so much.
All right, so because that was a pre-recorded talk, we won't have questions. We don't want to get sort of ahead of the schedule either, so we will just hang out for a few minutes before the next talk at 9:45. Tim, I don't know if you want to share the sort of rotating slide deck for the next few minutes, and we'll reconvene at well, we'll start up again at 9:45. All right, so we're getting close to the next talk. Tad, did you want to share your screen? We'll just get ready.
Sure. Thank you. It's disabled. I cannot.
Oh, really? Let's
This
see.
host has disabled participant screen sharing.
He's not yet co-host. Maybe this will.
Oh, that's right. Yeah, sorry, I was looking at the wrong person. All right, do you want to try that now?
Yeah. So, no, so I have some time, right?
It is 9:45 now, so you're welcome to go ahead.
Okay, thanks a lot. Majority of terrestrial animals are holometabolous insects, whether you tell it by species number or biomass. So it's legitimate to ask what made them so successful. And the key innovation of holometabolous insects is the complete metamorphosis, which means that the adult, the organs and structures of the adult organisms are not developing based on their larval counterparts, but they develop de novo from stem cells and other dissociated undifferentiated cells during the process of metamorphosis. And it has been argued that this developmental discontinuity decouples the larval adult phenotype, allowing them to evolve independently from each other. And that, of course, is to a large degree true, because a caterpillar is obviously very different from a butterfly, not just the way it looks, but the way it functions, what it is, and its physiology. However, it's also interesting to ask whether this decoupling is complete, or are there actually some genetic evolutionary constraints on independent evolution of larval and adult phenotypes, and I'm particularly interested in physiological phenotypes. So we've addressed this question not in monarch butterflies, but in another beautiful orange insect with dark stripes, Drosophila melanogaster. And we have been carrying carrying out long-term experimental evolution project, whereby 6 replicate populations are exposed to every generation to a very nutrient-poor larval diet. But once the adults develop, the transfer to standard diet and eventually fed ad libitum is to promote egg laying. And in parallel, cis-control populations of the same origin from the same gene pool are maintained on standard larval diet and otherwise treated the same way as adults. So this nutrient-poor diet creates obviously strong natural selection. It has resulted in adaptation to this poor diet in the selected populations. They're going to be always in red, whereas the controls will be always in blue. So they have improved their egg to availability on the poor diet, and they have evolved a faster development and also faster growth rate. And there's a very strong imprint of this adaptation in the genomes of those populations. They clearly separate along the first axis on the PCA on all SNPs in the populations. And you can also see this in larval physiology. Here summarized on PCA on larval metabolome, where especially among those raised on poor diet, the selected populations clearly separate from the control. So this not unexpectedly, this exposure to the poor larval diet resulted in strong adaptation of larval physiology. But we may ask, okay, what has happened with the adult physiology? As I've said, the adults are in fact exposed to the same kind of nutritional environment and aspects of the cycle. So first prediction one might make is, well, one might say, okay, the environment is the same, so there shouldn't be any different selection between the two regimes, so therefore we shouldn't expect any difference between selected and controlled adults. On the other hand, every generation, the selected adults, the selected populations, they develop on the poor diet, so possibly there has been selection on the adults to compensate for whatever negative effects of being food deprived in the larval stage might have been. So in this case, we would expect there will be some divergence in their physiology, even though they are on the same adult diet. But we wouldn't necessarily expect these changes to be similar to the ones we observed between the larvae. On the other hand, if the evolution of gene expression and therefore other traits that depend on gene expression between larvae and is not decoupled between larvae and adults, then we would expect that the selection acting on larval gene expression would have had consequences through pleiotropy, would have similar consequences for adult gene expression and metabolism. So to address these alternative predictions, we've exposed both types of populations to this poor larval diet and then the standard adult diet. And then we performed RNA-seq to look at global gene expression and also metabolomics on the fat body, which is the key metabolic organ of the flies or the insects. And we compared these results to data from another experiment in which we did the same on third instar larvae from both types of populations. So here are the results, the summary of the results for gene expression. So each dot is a gene, and the ones in purple are those that differ in expression between the evolved and selected and the control populations for both larvae and adults. And what you see in general, that there's a positive correlation, whether you focus just on those significant ones or on all genes, the genes that tend to be more expressed in selected larvae than in control larvae also tend to be expressed more in selected adults than in control adults. Similarly, for the metabolome, we see a positive correlation between the difference between the response to selection measured in the larvae in terms of metabolite abundance and the same metabolites in adults. Okay. Furthermore, as you can see by looking at the axis, at the scale on the axis, the scale is similar, and the dots seem to be spread in both directions similarly. So that implies, and we have actually quantified this, that generally the distribution of effect size is similar in the larvae and adults. So that means that even though the dietary selection was occurring at the larval stage, we see changes in adult gene expression and metabolome, which are similar to magnitude to those observed in the larvae, and the two are positively correlated. So there's positive correlation between the life stages. Now, one might think, especially for metabolome, that maybe, okay, well, you see the same difference or similar difference in adults as in the larvae, because these metabolites actually accumulate at the larval stage and they kind of linger. So whatever difference is in the larvae, you would expect in adults. So this is not the case, because if we look at the phenotypic plastic response of metabolome to the larval diet, so we expose whichever population to either poor or standard larval diet, and then do the same thing and look at their metabolites and compare larvae and adults. First, we don't see a correlation between the difference with the effect of this diet in the larvae and in the adults. Second, the variation, the magnitude of responses in the adults is generally tend to be smaller than in the larvae. So the phenotypically plastic responses of metabolome to larval diet are smaller in adults than in larvae, and the two are not correlated. So what we see for the evolutionary genetically based difference between selected control populations is not simply kind of mechanistic result of carryover effect from the larvae. So of those three alternative predictions that I've made at the beginning, the data seem to suggest the third one. And that is consistent with the idea that gene expression in adults and larvae is not decoupled from each other. And therefore, maybe these changes to be observed in adults are not actually adaptive. but they represent correlated negative or trade-off correlated responses to selection on larval physiology. So we looked at consequences of this selection on poor larval diet for adult fitness. Essentially, we again raised both types of populations on poor larval diet with the standard diet for adults, and then we look at their fecundity, and specifically in the time window at 24 hours in which during the experimental evolution, the eggs for next generation are collected. So this is the closest to adult fitness as we can get in this system for females. So these are exactly the conditions under which the selected populations have been maintained for last 17 years. Whereas for the control populations, it's a new situation. So based on that, you would expect that these selected adults will be doing better than the controls. This is not the case. In fact, we see that even though the control larvae, they do poorly on this poor diet. They die a lot, they take very long to develop, they grow slowly. But if they make it to the adults are doing better than corresponding adults from the adapted selected populations, and in terms of fecundity, and this is not due to a difference in number of ovules, which is a character determined during the larval stage. Also, the selected populations as adults are more susceptible to starvation. They die faster when deprived of food, even though as larvae, they're actually more tolerant to nutritional stress. So it seems that as a consequence of the adaptation of the selected populations to the very strong selection imposed by the very poor larval diet, the adults pay a cost in terms of their performance. So, and the fact that these changes in gene expression and the metabolome are quite robustly positively correlated, that suggests that actually in the short term, in this specific experiment, the gene expression patterns, larval and the physiology of adult and larvae are not completely evolutionally or genetically decoupled from each other, and therefore there is some constraint on the independent evolution of larval and adult physiology, even in the species in which the two stages are developmentally disjunct. Okay, thank you for your attention.
Great, thank you so much for a really excellent talk. There is a question in the chat for you.
The chat. Okay, I have to find the chat.
Here, I can read it out for you.
Yeah, please do.
So Annika says, great presentation. Did you collect data at any time points during the 230 generations of evolution? If so, when did the populations begin to diverge? Do they continue to diverge over time, or have they reached a plateau?
So for things like metabolome gene expression, we did not collect multiple time points. However, we did for measures of their performance as larvae, and they evolve very fast. And then, as you would expect, the thing has slowed down. Of course, it's a bit difficult to compare data obtained at different months or whatever, but I think they mostly have reached the plateau as far as adaptation is concerned. But there might be still, of course, there's still drift going on and inbreeding. And if this had been C. elegans, we would have frozen them and that way frost, but this is Drosophila, so we continue the experimental evolution. But the response was pretty fast initially.
Great, thank you. All right, so Adam, are you popping up because you have a question? Here, I can turn your mic on.
Yeah, thanks, Alex. Yeah, I just wondered, Tad, about body size. I may have missed it in your talk, but your data seem to make sense for the stress resistance and fertility if the poor diet animals are prioritizing survival over size, and therefore you're getting fewer of the larger standard diet ones, and they're bigger and more fecund.
And that's a sort of, I think, for a different reason, but the selected adults are slightly smaller, and that's probably have to do with the fact that they've been favored to complete development with less resources. But however, what's interesting in their metabolism, do you still see my slides? Yeah. So if you look at their metabolite abundance of, so we have done another experiment in which we compared just for the control populations, the flies that are in normal fed condition with flies that have been deprived of food for 24 hours. So that is on the x-axis, in the difference is on the x-axis in this graph, but and on the y-axis is the evolved difference in the fed state between select control populations in the last stage. And for several categories of compounds, in particular for amino acids, there's a positive correlation between what this suggests is that, or implies, is that the selected flies metabolically look as if they were already starving, even if they are fed. So I'm not sure that this fits your idea, but there seems to be something like that here.
Great, thank you so much. And if there's further conversation, we can continue that in the chat or the Q&A. And we'll move on to the next talk. So thanks again, Ted. And our next speaker will be Sophie Louise Heidig.
Hi, everybody. Can you confirm you hear me and you see my slides, please?
Yeah, yes, you're all good.
Great. Hi, everybody. Thank you for joining me in this session and also my talk on investigating evolutionary trajectories of proteins in marine Synechococcus. Now, marine Synechococcus are one of the most ubiquitous marine cyanobacteria. They're one of the primary producers in the ocean. They're globally distributed and therefore experience lots of different environments that differ in pH, in salinity, and most obviously in temperature. Now, these different environments expose them to a somewhat constant pressure to evolve, also on the molecular level. The environment affects all levels of the cell metabolism, in some cases transient, like the expression of heat shock proteins, and sometimes other changes get permanently recorded in the genome sequence as mutations. Especially for protein sequences, proteins being the workhorses of the cell that do a lot of the activities that a metabolism consists of. Simply exchanging one amino acid for another can potentially render a protein unfunctional. However, proteins are also known to tolerate quite drastic changes in their sequence. We know this especially for old proteins that have been present across all kingdoms, that they have or may have very low sequence identity, as low as 20%. However, the proteins fulfill the same function and they adopt the same three-dimensional shape. So for the data set that I'll be demonstrating my work today on is from the Cyanorak information system. It's a database specifically for Synechococcus and Prochlorococcus sequences. They are well annotated, mostly complete, and therefore lend themselves to extensive investigations in the interplay of ecology and evolution. Since my methods are applicable on an entire proteome scale, but today I would like to just show you the case of the small heat shock protein HSP20. A heat shock protein is a regulatory element in the temperature response of many organisms, also Synechococcus, and specifically heat shock protein 20 is conserved in Synechococcus as well as across kingdoms. It is also functionally highly conserved, so it's a very interesting example here. I would also like to point out that proteins can evolve differently from their organism. So it's not at the same rate and also not in the same way that protein sequences change in relation to the organism. You can see here two phylogenetic trees that I created with IQtree with good bootstrap support. And on the left, you can see a classical species tree that I created using housekeeping genes. On the right, the tree is purely based on HSP20, and different branch colors indicate different splits, which can be also highlighted in this way, where from the species perspective, the golden clade and the red clade are much closer connected than the golden and the blue clade. However, from the perspective of HSP20, the opposite is true. That leads me to some very interesting predictors that I want to integrate in this sort of phylogenetic workflow that investigate properties of proteins and protein sequences that are not as obvious from the sequence. So the sequence holds information, the evolutionary information, but it has been traditionally quite impossible to tell from this sequence what the 3D functional shape of the protein will look like. There's been drastic changes in this field in the last couple of years, where we now have AI-based predictors that can predict this final shape. However, they share mostly the same drawbacks, that they only show static final folding structures, but not the in-between steps. Those in-between steps are guided by local biophysical interactions that are transient, and therefore you have to consider them a bit more as a realm of possibilities of biophysical behaviors. And then the 3D shape would just be an expression of this biophysical behavior. These biophysical behaviors also tend to be more conserved than the sequence, and therefore I endeavored to apply this structure information that is now easily available to my data set of HSP20 proteins in Synechococcus as a basis for a multiple sequence alignment to make phylogenetic investigations. With this, I created a tool called SimSaPiper that uses structure information to make multiple sequence alignments, can even collect or predict structure information automatically. So if you're working on a protein data set, you might want to check it out. I created this multiple sequence alignment that you can see here, where you can also spot that while the sequence changes, the secondary structure information is very conserved, highlighting the usefulness of said information. And now I would like to take you through the biophysical property predictions. This here you can see is a prediction on beta sheet propensity of a single sequence of HSP20 using Dynamine, which is one of the in-house tools of one of my research groups that I am a part of. And you can see here in these peaks that there is 9 peaks here that indicate a high propensity for these residues to be involved in a beta sheet. However, when you look at the 3D structure down here that I have predicted, you can only count 7. Now that's an interesting discrepancy, especially for this case here, where the model structure doesn't show a beta sheet. However, when I compare it to other known examples of HSP20, there is experimental proof that in a dimer, this loop can form a beta sheet. So now this makes it interesting to investigate it in the full data set of all Synechococcus proteins, which would then look like this. The predictions are here aligned using this multiple sequence alignment that I created before. And in order to be able to connect this information with ecological information, such as the temperature at the sampling site, averaged over 10 years in this case, I have grouped the same plot here into distributions per range of temperatures of the sampling site for the organism. And you can see there with the orange arrow that there is an interesting peak for the high temperature adapted Synechococcus proteins, where one can now hypothesize if there is a functional implication here, for example, that this beta sheet is important to stabilize this active conformation of HSP20. I'm currently working on applying principal component analysis to this data set in order to quantify and to establish significances in my comparisons. And in order to achieve my main goal, which is using ortholog proteins to disentangle the influence of common ancestry from the influence of the environment and environmental conditions on evolution. With that, I would like to thank my colleagues at my both research groups, also my 3 supervisors, and the FNRS for funding my PhD thesis, and leave you with my take-home messages, which is 1, structure-informed alignments are really useful for phylogenetic investigations, and 2, biophysical behaviors should be considered if you want to get a better understanding of evolution. I am open to take some questions now, and if you have more questions, I'll also be with a poster at the conference in July in Montreal.
Great, thank you so much for a great talk, Sophie. Do we have questions? We've got lots of time.
Oh, indeed.
I mean, I'll start off. Okay, structure-based alignments. Are there particular situations in which they're going to be most useful, sort of for very old divergences or very rapidly evolving? Like, where do you think they're most useful?
They're generally most useful whenever you're investigating proteins. It doesn't really matter if it's ancient differences. It mainly relates to an interplay between sequence conservation, so whether the sequence is very similar. The influence of a structure-based alignment will be much higher if you have very dissimilar sequences. They're also very useful if you're not looking at globular proteins. So lots of proteins have a kind of round shape, and this is what all the alignment algorithms are predominantly trained on. So they're a lot better for these kind of proteins. But if you're working with membrane proteins, they tend to perform quite suboptimal. Their structure-based alignment is helpful.
Gotcha.
And yes.
Question, which is going a bit away from the structure, but I mean, this is super useful for structured proteins, but a lot of proteins in many genomes of many species are unstructured. Do you think you can develop something similar more from a functional perspective? If you look at let's say you have a disordered structure, and you just look into the biophysical properties of these disordered sequences, and then use this information.
Indeed, this is one of the further goals that I have set for myself in my PhD, where this information is very useful, but right now I don't have concrete plans to do that. However, it's really informative to estimate how well your alignment currently works. There's also, I can account for a disorder in the latter half of my project, whereas the first step is to create a multiple sequence alignment. But I have also, like the beta sheet predictor, there's predictors for disorder that I can use and then account for if this alignment works really well. But indeed, it's one of the weaker points of the multiple sequence alignment pipeline is when you have a disordered region, there is no structure that you can use to align.
Yeah, I'm working with this. I totally understand the struggle.
Yeah, I have another question, but does anybody else have a question before I jump in? Okay, so when you showed the sort of mismatch between the phylogenies, the sort of the species tree and the HSP20 tree, and when I see that, I think recombination.
Yeah.
Are there other things you think are going on there, or is it just a matter of recombination between lineages? Yeah, what do you?
The sequences that I show here, they are not locally close.
Okay.
The samples that I show here are from all across the globe. So while it's not impossible, it is generally, I think, not the main thing that is acting here. What I tried to show here is that there is proteins tend to have their own system of evolutionary pressure. There's proteins that are sort of fixed, that rarely change sequence, and then there's other proteins that evolve a lot. When there is, for example, gene duplication, you would also see quite a difference in this tree. which is super interesting. So I don't believe it is mainly recombination. I think it's more like the gradual shift of mutation and then selection based on mutation. So one of the things that I work on integrating is using tools like PAML, if you're familiar, to identify regions of, like, per residue, to identify regions of under evolutionary pressure. That also works on like the entire tree, but I understand that in order to use PAML to work on the entire tree of proteins, you're supposed to have distantly related organisms, and mine are the opposite of that. So I think it won't be optimal to use here. Well, I'll investigate it.
Great, thank you so much. And good luck with your future investigations. And thanks again for your talk. I don't see our next speaker in the room. Shabani, are you here? Can you give us a sign in the chat or raise a hand? Let's wait a minute or two. I don't want to sort of prematurely call things to an end if they're just running late. Give me a second. I'm going to pop into the back rooms here. Oh, Tim, you're already looking into that. Thank you.
We are on our title slide. The talk is indicated. I think there was another list where it was not there, so I'm not
okay.
absolutely sure it will take place.
Okay, gotcha.
Um,
So their talk isn't on the title slide, is what you're saying?
yeah, so
Okay.
it could be here, but I agree that the name is not to be found.
Yeah. Okay. One more time, Shabani, are you here? Okay, well, while we're all here, if anybody has questions for any of the speakers today or discussion points, yeah, might as well use this time. Sure, there's just some chat going around here. All right. Well, thank you everybody for being here. I really enjoyed the talks this morning, and I hope you have a good rest of the day. Enjoy the rest of the virtual meeting. So, I mean, I'm going to stay here in case our last speaker does arrive, but following that speaking slot, we're on a half-hour break. So the next set of sessions start at 11 Eastern time. So thanks again, everyone. Really enjoyed that set of talks.
Bye.
Bye. Thank you.